In this session, we will cover extensions of the phylogeographic models. The models presented in the previous section lead to ancestral reconstruction, drawing statistics, and visualizations with the objective of reconstructing the past. Although useful, it remains difficult to test hypotheses with them. This is why developments have recently been moving towards hypothesis testing. The question now is, which processes gave rise to the pathogen evolution and dispersal? This new generalized linear model is how we test predictors of spatial patterns. Predictor can be, for instance, geographic distance, assuming that nearby locations undergo more diffusion than those further apart. The generalized linear model is achieved by making the log of every movement as a log linear function of several potential predictors. Here, beta coefficients allow the data to estimate the contribution of the predictor, or how big they are, and the binary deltas, which predictors should be included in the model. The first study where this model was applied was to understand which factors were determinants in the evolutionary and spatiotemporal history of seasonal influenza H3N2. Here, several predictors were tested, such as distance, air traffic measured by passenger flow, population density, among others. As expected, air traffic was estimated as the most relevant predictor determining H3N2 spread. This finding is not surprising since air traffic is, of course, an obvious means for pathogens to rapidly spread globally. But it does serve as a proof of principle for the GLM approach and the fact that genetic data can capture such information. As covered in the population structure session, in epidemiology, what we see may be interesting, but what is concealed is crucial for the more realistic reconstruction possible. This was the case particularly at the beginning of the COVID-19 pandemic, when the number of viruses sequenced was a small fraction of the number of reported cases. Spatiotemporal bias in genome sequence sampling can severely confound phylogeographic inference based on discrete trait ancestral reconstruction. This has impeded our ability to accurately track the emergence and spread of SARS-CoV-2, which is the virus responsible for the COVID-19 pandemic. The missing data were problematic due to the severe undersampling of particular regions, particularly those reporting high numbers of cases such as Italy, Iran, and Hubei, China. So, to account for the missing data, molecular epidemiologists thought of two strategies. The first was to get more data besides genetic data. This is achieved by incorporating travel history into phylogeographic inferences. This is useful to gain insight into the viral diversity from undersampled locations by including the movements of travelers arriving from these locations. Using the GLM extension we just covered, we can also incorporate air travel flows as predictors of potential viral introductions, which is useful to model early outbreaks before travel bans. The second strategy relied on simulating genetic data by questioning the probability of a tree topology under different outbreak scenarios. We can also include unobserved sequences in phylogenetic inferences based on high case counts in addition to the available sequences. With the unprecedented accumulation of genomic sequences from across the world comes a vast collection of metadata on individual patients, such as in some cases details on their recent movements like travel history prior to having developed any symptoms. This information can be explored in order to yield more realistic reconstructions of the virus spread, particularly when travelers from undersampled locations are included. When the sampling location of a sequence is available, the tip state for that sequence in the phylogeny can be set to that location. This corresponds to the classic discrete phylogeographic inference scenario that we covered initially and is typically used in the absence of travel history information. The result of this is shown in the first tree. There, using only sampling location results in an unrealistic Australian ancestry and two transitions from Australia to Italy, likely because Australia is represented by the largest number of tips. Lamey et al. Proposed to accommodate travel histories by augmenting the phylogeny with ancestral nodes that are associated with the location state, but not with the known sequence, and hence enforce that ancestral location at a point in the past of a sequenced sample. The second tree illustrates the concept of introducing ancestral nodes associated with locations from which travelers returned. The ancestral nodes are indicated by arrows for 5 cases relating them to the genome sampled from the travelers and are introduced at different times in the ancestral paths of each sample genome in order to inform the reconstruction that at this point in time the lineage was in that location. The upper arrow in the second tree represents the information introduced for the traveler that returned from Hubei to Italy. From top to bottom, the same procedure is applied to the 4 genomes from travelers returning to Australia from Hubei, Iran, Southeast Asia, and Hubei again. The result of this inference is shown in the third tree. Incorporating travel history in the discrete phylogeographic inference yields more realistic reconstructions of viral spread compared to other available approaches, such as the default discrete phylogeographic model. Phylogeographic inferences were further confounded by the relatively low availability of genomic sequence data from locations that experienced early outbreaks, including Italy, Iran, and the original epicenter in Hubei, China. In Europe, a narrative took hold that the virus in Germany had not been contained, but had been transmitting undetected for weeks and had been carried out to Italy by an infected German. Werby et al. Tested this hypothesis by developing another extension of the discrete phylogeographic model that incorporates unobserved sequences with the aim to account for the effects of undersampling viral genetic diversity in the epicenter of the pandemic. Phylogeographic reconstructions are highly sensitive to sample bias, and in this case, 12 locations were identified as critically undersampled by contrasting the number of available genomes against the cumulative number of recorded COVID-19 cases. So, for poorly sampled locations, they included unsampled genomes in the Bayesian inference that were associated with an epidemiological informed sampling time and location, but not observed sequence data. This shed light that both the Italian and German outbreaks were caused by separate introductions from Hubei, and that the particularly impressive response to an early outbreak in Germany delayed local COVID-19 outbreaks by a few weeks and brought crucial time for the US and European countries to prepare for the virus when it finally did arrive.